Good morning, everyone. Today I'm going to make a small summary of the research article titled Reducing Ex vivo Culture Improves the Antileukemic Activity of Chimeric Antigen Receptor CAR T cells. So, basically, despite the high overall rate of complete response to CD19 specific CAR therapy in acute lymphoblastic leukemia, some patients still relapse due to premature loss of the engineered T cells. T cells undergo progressive differentiation during ex vivo culture that is associated with reduced persistence following adoptive transfer. Maintenance of the less differentiated states depends upon soluble factors and cell-cell interactions found within tissue microenvironments. Extensive ex vivo culture of T cells, although amplifying T cell number, also leads to significant loss of stimulus that limits the engraftment and persistence following adoptive transfer. So, with this in mind, this paper demonstrated the beneficial effects of reduced culture duration resulting in improved in vitro proliferation and effector function of CAR T cells. So, for one of the first experiments that they developed was to study primary human T cells differentiation over time. So, flow cytometry was used to distinguish different subsets of cells. First, they distinguished basically the T cells specifically, and then the cytotoxic and inhibitory part of the T cells. Then we can see here the four different subsets that we have. First, we have the naive T cells here, then the central memory T cells, the effector memory T cells, and the effector T cells. And also inside the naive T cells, they also were able to recognize two types. The stem cell memory that we have here, and also the naive T cells properly done. So, also we can see here, like in one of the first analyses that they did in the initial count, basically the T cells from the leukemic patients exhibited a significantly greater proportion of effector differentiated T cells, as we can see here, and compared with the healthy donors, being the subset representing the majority of the population. In the other way, we can see here like the naive T cells comprise a proportionally lower percentage of the total T cells compared with the healthy donors. So here is our first difference between the T cells obtained from healthy and for leukemic patients. Following the activation through the TCR-CD3 complex and the CD28, the T cells entered a phase of rapid proliferation that was associated with the progressive differentiation during 9 days of ex vivo culture. Here we can see, again using flow cytometry, how the subsets move around the different quadrants of different subsets. Now, analyzing just the healthy donor cells, we can see how these cells underwent an approximately 30-fold expansion during this 9 period of culture. And analyzing the proportions of T cells, we can see how they underwent a shift from the naive to a central memory immune phenotype. Here at the beginning we have more naive T cells, but at the end of the culture, which was 9 days, we can see that the central memory cells and the effector memory cells are the ones that are in the highest number. In the other side, we then have the analysis of the leukemic patient cells, and here we can see how the cultures derived from patients show a reduced expansion associated with an initial decrease in total viable cell number. In the case of the analysis of the phenotypes of the cells, we can see how in this case, the central memory T cells showed a relative increase in frequency in cultures, and basically all the phenotypes was similar to that observed on healthy donors. And at the end in the day 9 of culture, we can see how the final T cell population was substantially more differentiated compared with the healthy donors. We can see here how in the case of the leukemic patients, their cells develop effector memory cell in higher numbers than the healthy ones. Taking all of these Results from this experiment, they conclude that the T cells progressively differentiate over time during ex vivo culture. Next to this, we pass to another experiment. They basically develop a systematic analysis of T cells harvested at various time points, beginning at day 3. Day 3 basically is 72 hours after the anti-CD3 CD28 stimulation and 48 hours after the lentiviral transduction. This day was chosen because it was the earliest time point at which gene transfer could be robustly evaluated. For this experiment, they used the chromium release assay to check the cytotoxicity of T cells. Here we can see, like in image A, how the early harvested CAR T cells show a consistently enhanced cytolytic function compared with more extensively cultured T cells. In this case, we have in blue lines the harvested cells at day 3, and in orange cells the cells harvested at day 9, and in green the control cells. Also, they tried different ratio of effector target cells, and we can see like how the more effector cells we have, the more specific lysis we will have, and also how the harvested at day 3 cells are more efficient compared with the same cells harvested at day 9. Also, here, they perform an independent study using another patient's cells, and the specific lysis was also higher in the cells harvested at day 3. In the case of the early harvested T cells in day 3 and 5, derived from healthy donors, they show a significantly enhanced proliferative capacity following antigen exposure. In this case, we can see here in the day 3 and day 5, they have higher fold increase. But comparing with the cells used from leukemia patients, the ability to produce effectors, the T cells from these patients did not show a demonstrable difference in proliferation between day 5 and day 9 harvested cells. Next, the ability to produce effector cytokines in response to antigens in T cells derived from healthy donors was also checked, and we can see how it is unaffected by time. Independently of the day that they were harvested, the production of cytokines are the same. In this, taking all of this into account, the conclusion of this part of the experiment was that early harvested CAR T cells exhibit enhanced effector function and proliferation. For the next experiment, they evaluated the potency of CAR T19 cells derived from healthy donors harvested at different days but in vivo. For this first, using mice, they were injected with leukemia cells, and after 5 to 7 days, they were injected with the CAR T19 cells harvested at day 3, day 5, day 9, or also our control cells. We have to mention here that these CAR T cells were also marked with the luciferin, so they can perform the luciferin-luciferase system. So the total flux of bioluminescence was measured in order to check how was the cancer control developed between these different type of cells. Here we can see that also they tried two different doses of CAR T cells, and we can see how these cells demonstrated the antileukemic activity. A higher dose of T cells, here in image B, exhibited durable efficacy in the majority of mice, as expected. However, in the early harvested cells, day 3 and day 5, they exhibited superior tumor control compared with the cells harvested at day 9. When using a 6-fold lower dose of CAR T19 cells, the efficacy was lost compared to the cells harvested at day 9. Also, as the control, we can see here how the disease progression in mice treated with the control cells was developing. Next, we can see here how only the day 3 harvested cells exhibited cell dose persistent leukemia control in all mice at the lower doses. And in the case of the higher doses, the harvested day 3 and day 5 cells, they have at the end of the experiment, they have this persistent leukemia control. What's more, they also here evaluated the persistence of T cells. So for this, the absolute counts of CAR T19 cells in the peripheral blood of mice were taken for the experiment. And we can see here how the control of leukemia was associated with the improvement persistence of T cells, demonstrated by the significantly increased absolute counts of CAR19 cells in the peripheral blood of mice. Here we can see, for example, at the end, in the 38, that the day 3 harvested cells are in a bigger quantity compared with the other ones, which is associated to the proliferation of the control of the proliferation of cancer. And at the end, they also, with the harvested T cells, demonstrated they use only an analysis of the day 5 and day 9 cells, and we can see how there's a greater production of cytokines in vivo by day 5 harvested cells compared with the day 9, which was consistent also with their improvement in the antileukemic function. Taking all of these into account, we can see also how the early harvested CAR T cells exhibit enhanced potency and persistence in vivo. To conclude, the last experiment that they developed was to evaluate the clinical feasibility of an abbreviated culture approach. So they performed a comprehensive retrospective analysis on the CAR T cell products generated from 27 patients undergoing treatment for relapsed or refractory leukemia. Here we can see in figure A how the patient's T cell underwent logarithmic expansion in response to beta activation. And in B and C, we can see how it is shown like 70% of the day 3 products and more than 80% of the day 5 products will meet the pre-existing day 9 release criteria with respect for cell viability and also with respect to dose. And here we can see how they pass the threshold to meet the clinical dose expected. Taking all of this into account, we can see how, in summary, these results demonstrate that culturing CAR T cells for a shorter duration yields a cellular product with less differentiated cells, significantly enhanced effector function and enhanced proliferative capacity. Collectively, these features led to enhanced antileukemic efficacy, checking potency and persistence in vivo. In retrospective analysis of CAR T cells, cell harvested at day 3 meet the existing day 9 criterion for achieving infusion criteria in 70% of all leukemic patients, further supporting the feasibility and immediate translational potential of this approach. Also, minimally cultured T cells, therefore, provide a simple and more cost-effective method for achieving potent CAR T cell immunotherapy and deserves evaluation in the clinical testing. Thank you.